Hi and good morning, everyone. Thank you for this great opportunity to present with you all one of our many surveillance projects. I will present on behalf of our team on zoonotic arbovirus vector surveillance and molecular detection in Papua New Guinea. In Papua New Guinea, there is limited knowledge about Japanese encephalitis virus and other zoonotic arbovirus, especially regarding how common they are, where they occur, when they spread, what strains exist, and which mosquitoes carry them. Our study was conducted from 2019 to 2024 at 14 agricultural farming sites in 4 provinces in PNG: NCD, Central, Morobe, and Western Province. Our study was done in sentinel surveillance sites for the PNG National Agriculture Quarantine Inspection Authority. Our longitudinal surveillance was done in 2023 and 2024 in Central and Morobe provinces. Our surveillance efforts and methods have thrived under the One Health approach, with PNGIMR as our principal cross-sectoral partner. Most sentinel sites operate under NACIA's animal health surveillance sites, and we work together with both NACIA's animal and plant health teams as well as Department of Agriculture and Livestock in provinces like Western and North Fly. We coordinate closely with NACIA for scheduling, logistics, and farm and community engagements, and they have been a great support in transport, farm access, and facilitation with farm managers. In areas where IMR lacks office facilities such as Morabe Province, Nakia provides workspace or laboratory access, and Morabe PHA has also allocated a small operational space for us to carry out our on-site surveillance activities. In Port Moresby, we have a mini insectary at the Nakia Kila Kila, where we use it for most of our vector surveillance work in palm. Nakia has played a key role in supporting our field work, primarily by providing vehicle and driver assistance as well as officers who join our teams on site. The logistical support has helped us reach remote farms where surveillance activities are conducted. Beyond the transportation, we work collaboratively with NACI, exchanging knowledge and strengthening our shared capacity. In locations such as Western Province, we have been linked with the DAL, whose team has supported us in our surveillance work. Similarly, we've built working relationship with various PHAs that we have worked in their province, especially in Morobe province with their PMS and the team, which has enabled resource sharing. From a technical perspective, our team benefits from involvement of cross-trained officers with expertise in entomology, epidemiology, and animal health. We also share laboratory equipment and workspace for arbovirus testing and other specialized assays. Our field surveillance method. So in the field, we set up adult mosquito traps at the piggeries. We have about 14 days of mosquito trapping per site or per piggery. So after we set that mosquito traps, we collect the mosquitoes every day for the next 14 days. So when we collect the mosquitoes, we knock them down for about 10 to 20 minutes in the freezer. Then we sort the collections by separating the mosquitoes from the non-mosquitoes, because when we collect our catch packs also come with non-mosquitoes. After we separate them or sort them out, mosquitoes from non-mosquitoes, then we discard the non-mosquitoes, and the mosquitoes, we identify them using microscope to species level. And they are blood meal, as in unfed, partially fed, and fully fed. And then we store them in RNA later, and then the mosquitoes are brought to the lab for processing. At the lab, we pull the mosquitoes again by the species, blood meal, according to the location of collection, month and year of collection, and the trap type used for collection. After that, we then homogenize the mosquitoes from the pools that we have pulled them. And then after we homogenize, we do RNA extraction, and then we run PCR. So most of our sample processing for this surveillance was done at the Mall Hub Partnership in Health Laboratory in Port Moresby and ACDP in Geelong. Here is our 2019 to 2021 mosquito species distribution. So from our recent publication in 2022 for this work, we compared the three different types of trap, the mosquito traps that we used in the poultry and piggery farms. So we compared the CDC normal light trap, CDC UV light trap, and the BGS trap, and found that the CDC UV light trap to be most effective in catching Culex specific mosquitoes in PNG farms. So from this, we collected a total of 29,920 mosquitoes from the 4 different provinces that we surveyed. And out of the 29,920 mosquitoes, 18,951 mosquitoes were Culex. In our recent longitudinal surveillance result, we conducted the surveillance in Morobe and NCD only, in piggeries only, and we used BG Pro UV light traps and BGS traps. So from these, we collected a total of 37,077 mosquitoes, and 36,710 mosquitoes were of the species of Culex. So as you can see from the results here on the graph, you can see that the BG Pro UV collected more mosquitoes than the BGS trap, and also more Culex mosquitoes. Our PCR results from 2019 to 2021 in Morobe, we detected 2 positive for JEV from Culex gelidus mosquitoes and 2 Cocombera virus from NCDO Central from Culex annulirostris and Culex quinquefasciatus mosquitoes. In Western and Northern Province, we also got positives for Cocombera virus, and they were found in Culex pilus mosquitoes and Culex quinquefasciatus. So in 2019 to 2021 surveillance, we detected a total of 2 JEV and 5 Cocombera virus from the 18,951 mosquitoes that we collected. Our PCR results for the recent surveillance from 2023 to 2024, we detected 9 JEV positives and 2 Murray Valley encephalitis virus positives from Morobe province. So 7 Japanese encephalitis virus from Culex gelidus mosquitoes, which were like a continued detection from the first detection in 2019 to 2021. And we got the first JEV detection from Culex quinquefasciatus and Culex citians, still from Morobe province, and we got 2 JEV positives from NCD or Central from Culex gelidus mosquitoes. So altogether, we detected 11 JEV positives and 2 Murray Valley encephalitis virus from the mosquitoes that were collected from 2023 to 2024. Here is a phylogenetic tree showing the JEV sequencing for the 2 JEV positives from 2019 to 2021. So the JEV genotype belonging to the genotype 4 were confirmed from Culex gelidus mosquitoes collected from the piggery in Ley from 2019 to 2021. So the JEV genotype belonging to genotype 4 is the first detection in PNG, and it is the first time this genotype 4 has been detected in Melanesia, Australia, and New Zealand, as previously it was genotype 1 and genotype 2. So the phylogenetic analysis revealed that JEV detected in PNG samples clustered with a recent large-scale JEV outbreak in Australia in 2022. In summary, our study presents the first detection of JEV in Culex gelidus mosquitoes from 2019 to 2021 and continued detection of JEV from Culex gelidus from 2023 to 2024. It is the first time an arbovirus has been detected in zoonotic arbovirus vectors in PNG livestock farms. Our 2023 to 2024 surveillance showed continued circulation of JEV, and also the first detection of JEV from Culex gelidus mosquitoes from Central Province. Also a first detection of JEV from Culex quinquefasciatus and Culex citians, and the first detection of Murray Valley encephalitis virus in Culex gelidus mosquitoes in Papua New Guinea. Our study highlights the biosecurity and public health risk that JEV and MVEV poses in PNG, and especially in communities of our sampling locations, and it highlights the importance of a One Health approach with PNG cross-sector and regional collaboration to facilitate surveillance and control of cross-border zoonotic pathogen threats. Thank you. Time travel with us in this 1 to 2 minute slide of pictures showing our work in the field and enlighten the people that we have worked with from 2019 to 2024 from all the provinces, both for mosquito and human surveillance. And we acknowledge all who are part of our work and have worked behind the scenes to facilitate our successful work. We acknowledge all organizations who have worked with us and has been part of our surveillance work. Thank you. That's the end of my presentation.